ළමයි ඔයාලට ඇහෙනවද කියලා chat එකට message එකක් දාන්න. Can you hear me right? English medium students? Can you hear me? You can share the link, right? Share the link with your friends and you can just put a status or something, right? In order to notify the others, right? So it will be better because English medium paper discussion and such like things for A-level is such rare, I think. I reckon going through the YouTube also, right? If you scroll down top to bottom, there won't be such MCQ discussion or maybe English medium classes are very rare. So in that kind of a scenario, in that kind of a atmosphere, right, that we are conducting the English medium based classes, right? It means meanwhile, along with the Sinhala medium class, we are doing the English medium class also. All the tudes, all the papers, and all these stuffs and all those things and the theory explanations, then words in the resource book, and all we used, right, for each and every lesson. Meanwhile, going through with the Sinhala medium class, we are providing the English medium stuffs too, right? So there isn't any there is no any actually any separate English medium class up to 2023. We'll see what will happen in 2024 and 2025 likewise, right? For 2023, there is no such separate English medium class from myself, right? In that aspect, you can join the Sinhala medium class, but no issue. During a theory video also, you can go to my YouTube channel and you can see there in each and every lessons and each and every paper discussion. I refer to Sinhala medium. It means during the Sinhala medium discussion, I'm again discuss the same thing with English medium students at the same class. So today, here I am. It means I plan to conduct this session separately for English medium students in order to right it means to notify you. And there are so many students who are struggling with the biology, right? There are more than 50 days remaining. So you can actually cope on that. You can actually work on that and you can go to your dream of medicine or maybe your dream. It means actually from my point of view, you can get the A pass for the biology by doing the necessary steps as it means according to the way I'm talking. It means for an example, I'm asking you to do this thing, then you have to do it. If you skip those things, naturally you can't do, no, right? So if you go through the process, it means if you go through the rules and regulation and all those instructions I'm giving day by day, then if you follow those 100%, then actually you can achieve this in the remaining 60 odd days, right? So in order to it means to notify the students who are involved in the A level, advanced level, and there's a class like this, right? So we are doing these kind of stuffs, and we are doing quality papers. So all those things that are I need to just focus on you. That's why I'm today holding or just plan to do the English medium paper discussion separately. Now I think you have already completed the paper, right? I think you have already completed the paper, right? If anyone who don't have the paper with you now, right? You can get it from our Telegram channel, right? I will just post the link in the chat section, right? Okay, in the chat I will post the link. Okay, wait. Right? Yeah, you're asking where you can get the paper, right? So wait, and I will yeah. Now this is the Telegram group link. So I will pin it, right, in the message. Then you can go to the Telegram group, and there I will post the paper. There I will post the paper, right? No, actually I will yeah, I will put it in the group too, and I will put it in the live section also, right? Wait. You can get the paper and I have pinned it, right? Okay. You can get the paper from the relevant link, and link is there in the comment section, right? In which comment, not the comment, the chat section. So that is what we are having up to now. So now we'll start the discussion. Now this is the support paper, right? There are 50 MCQs. It covers the entire syllabus. So you will get the entire theory aspect and all the necessary knowledge related to the whole syllabus, right? Okay. If there is any particular issue, if there is a noise issue, or if there is a lagging or any such kind of a thing going on in the live, right? You can just chat in the live section and just inform me now. This is there's issue likewise, right? So then we'll start. Okay. Then send me the message as usual, right? English medium students, as usual, send me the message. Are you ready for the task, right? Now we have 50 MCQs with us. Now we have to have a struggle and have to complete it, right? Do you ready? Just drop a message in the live section quickly. Just drop a message, right? Are you ready for the session, right? I think you have to have that gut feeling, right? Gut feeling with you before or prior to a such kind of a session, or prior to write the paper likewise. So okay, same here. Okay, right. 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 Then we'll start the session, right? Okay. Now this is the biology support paper 1. MCQ discussion. Right. Okay. First MCQ. Which of the following indicates the hierarchical level of organization of living things in the correct order? I won't take far long time to discuss this paper, right? I will just mention you what is the aspect of testing, this aspect of theory which is tested in the question, and then we'll go around the discussion quickly, right? Okay, get ready. Okay, first one. Which of the following indicates the hierarchical level of organization of living things in the correct order? Actually, this is very easy. Now, from starting from the molecule level, you can get the molecules. Then it forms organelles. Then what? Cells. Then tissues. Tissues into organs. Then organ into organ and systems. Then again, it forms an individual, right? Okay. Then that individual get collectively. It means they are get collecting, and they form the what? The community, right? Not the community, it's the population, right? Then it goes into community, then ecosystem, right? Then it go into the biosphere, right? That is the order. So here, the entire the organization or all the organization levels of the hierarchy is not given. What is given is some 1, 2, 3, 4, 5 levels, and you have to, from the starting level onwards, you have to calculate, or you have to understand, or you have to choose the correct answer. Now, from the cell level towards, you have to get cells. No, not the organelles, right? From the organelle, cells are formed. So therefore, the first one is incorrect. You can clearly take first one as incorrect. That is the reason. Now, the second one, population, right? Community, right? Ecosystem, biosphere. That is correct. From the population onwards, you have to get the answer from the population onward, right? Okay. Then species, organ system, and population, right? So there. Now, species actually is not a hierarchical level of organization. In that aspect, you can't take the third one as the answer. Then species once again is given. It is not coming under the hierarchical level of organization. Then again, organ, again the species, right? It is not given, right? It is not given. No. It is given, right? It is after the individual. Then there can be species, right? Okay. Then, but according to it means the arrangement, actually it is not given as the hierarchical level of organization, but species is the collectively we can name that group of people. You can say they are the human species, right? Likewise, but it's not given as a a kind of a hierarchical level in the organization, right? Okay, that's the thing. So the answer is for the first one is second one. Answer for the first one is second one, right? Okay. Then, which of the following consists phosphodiester bond in its structure? Now, better to tell the answer. In the discussion, it is better, I think. Then the session will go very smoothly and softly, and you are involving in the session far more better. What is the answer for the second one, as you reckon? What is the answer for the second one? Yeah, what contains the phosphodiester bond in its structure? It has to be a what? It has to be a polynucleotide. Your answer, drop it in the chat, right? Drop your answer in the chat. So phosphodiester bond is found in a nucleic acid. It has to be a nucleic acid. It has to be a what? Ribonucleic acid or a deoxyribonucleic acid. So in the given answers, yeah, first one is mRNA. mRNA means what? It's a ribonucleic acid. It's a nucleic acid, right? So second question. First answer is actually correct. The first most given one is correct. Triacylglycerol is a lipid. So therefore, phosphodiester bonds are found in nucleic acid. No? In that aspect, second one is incorrect. Phospholipid once again a lipid, so that is incorrect. Fourth one, NADP, it's a nucleotide. So therefore, we can't find phosphodiester bond in nucleotides. We can find in nucleic acid, right? Nucleic acid is formed by the polymerization of nucleotides. No? So therefore, we can't get it. Then ATP2, it is a nucleotide. It's not a nucleic acid. It is not a polymer. It's a monomer. No? It's a monomer. So therefore, we can't find phosphodiester bonds, right? So the answer for the second one is first one. Right. Then for the third one, right? Before I'm expressing or I discuss, you have to tell the answer for the third one also. Drop the answer in the chat. Drop the answer in the chat, right? Drop the answer in the chat. Okay. Now we are getting back to the third one. The following statements describe three orders of structure of the insulin molecule, right? Insulin molecule, it's an what? Insulin is a what? Is it a enzyme? No, it is a hormone, right? Insulin molecule, they are testing. First one given, A. The molecule consists of two polypeptide chains joined and folded around one another. Now this is the critical point here. You can take a highlighter and then you can highlight two polypeptide chains in the question. It means more than two or more polypeptide chains means actually it is the quaternary. Up to the tertiary level, from the primary, secondary, then to tertiary, you have only one protein, like it means one polypeptide chain. So if it is more than one polypeptide, it means two or more, then it comes under quaternary. So for the third one, A has to be the quaternary. A has to be the quaternary structure. Then B. The sequence and number of amino acids in each polypeptide chain is linear. It means that is saying that the amino acids are arranged in such a way, in a linear way, that they are just one by one, they are connected. Now, that is the simplest level. It is the primary, no? It is the primary level that is discussing here. See, the amino acids in each chain are coiled into a helix and held in position by hydrogen bonds. Actually, the helix and the beta plated and the alpha helical, those structures are found in the secondary structure. Those are found in the secondary. So therefore, the answer for the third one is quaternary, primary, and secondary. It is given in the fourth one, right? Right. Then which of the following IP's objective combination will allow one to see the largest number of cells? Right. largest number of cells within one microscopic field from an epidermal peel of onion, right? So you have better to highlight the largest number of cells. It is asking the largest number of cells, right? There are two aspects. When we are observing through the microscope, there are two aspects. One is that we can see a particular thing which is enlarged, right? For an example, now here is an example, right? Now I am having a right, just a circle like this, and I have just separated this into segments, right? So here how many now? You can find 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 segments you can see. Now this is the real one, right? This is the real one. So we use a microscope in order to magnify, right? In order to magnify this, right? So in one occasion, if we need to magnify this particular one, if we it means I will just draw it like here. We can find the name what this is. Now here we can find here I have written something here in this square, right? And someone will get this notice, right? In the real image too. We'll see. Will you get it correctly, right? Guess what is there inside? There inside in the square. What is there? Inside the square, what is there? Is some 3 letters I have written, right? You can better to put it in the chat, right? Okay. Then, right? Okay. If we just magnifies it, then we can get something. I mean, if you need to just highlight or you need to get furthermore details of the relevant square or the relevant thing, right? Then you will magnify it, right? Now this is the same visual field. Now the visual field size is not changing, no? Right? The visual field size is not changing. It is the same thing. Now for an example, now you will take this square as like this. Now this is the view of the particular square. Now you are having this one in the left side and this square in the right side and the top one and the bottom one also, right? Now these are the 4 things, right? Now you can take now this one. These are the 4 squares which are neighbor to this one. So you can take this one, and this one, right? There are 4. Now you can just magnify and you can take a clear notice of the letter or the word or the phrase which is inside. That is the PMC, right? Okay, right? So that is one aspect, right? So then you are magnifying further and furthermore. It means if the magnification is too much, right? You can just go through one thing or one particular cell. If there are a number of numerous cells in a certain visual field, if you need to go through it means further clarification of a certain cell, to go through in detail of a certain particular cell, you can just magnify, right? It will give out a large view, right? So if you then what happened now what happened, you can just see now here you have 16 squares here, right? Now how many you can see? You can see only one, right? One here. So when what happened, when the magnification get, right, then the magnification get increased, what happened? The number of cells you can see get decreased, but the clarity of the certain particular cell increase. It means you can get the details of a certain particular cell you are expecting to watch. Got it, right? So you can just write down here. Now with the magnification, you can just derive just an equation or a right? So a proportion, right? The magnification is what? It's directly or inverse. It is inversely proportional to the number of cells, right? So when the magnification is then actually we can put it here, right? The magnification in this side is better, I think, right? So then the number of cells in the field, right? Number of cells in the field, right? So with the magnification, what happened? When the magnification is increasing, the number of cells are decreasing. When the magnification is very low, then the number of cells in the field is high, right? Now this is the equation you can derive from that, right? Got it, right? This is the equation. Okay. So that is regarding the equation, right? Therefore, which of the following IP's objective combination allow to see the largest number of cells mean with the lowest magnification? Now here in the answers for the question number 4, in the first one it is giving 5 into 40. it means it gives a magnification of 200. Then second one, 5 into 100, it gives a magnification of a 500, right? Then 10 into 10, it gives a magnification of 100. In 10 to 40, it gives a magnification of a 400. Then the other collection of lens gives a 1000 magnification, right? 1000, right? Likewise here, what is the lowest one? That is 10 into 10. So the answer for the fourth one is clearly and easily we can take for the answer for the third one, fourth one is third, right? It's the third. It's the third, right? Right, throughout the discussion, I will go entirely with English medium. The reason is now I'm somewhat proud to say that, right? There are some Tamil medium students, I just noticed them, right? There are some Tamil medium students who are also, it means those are doing the A-level in English medium, but actually there are some students who are doing in the Tamil medium and who are getting into our class, right? So it means who are fond of English, and there are some students who are Tamil, mother tongue is Tamil or Islamic, then they are coming to the English medium class, so they can't understand single. There are such students also. So in that aspect, if you are conducting a session English medium, I will go, the entire session will be based on the English terms, right? I won't use any Sinhala term. So don't be disappointed, right? If you are doing English medium and you are Sinhala, I think it means you might have the capability of having or the grabbing things in the English medium manner, right? Okay, right, here and there I won't use Sinhala medium terms, right? I think it is far more better, right? Okay, this is English medium now. Fifth one, the following statement describe few steps in the cell division. Right, we'll go. Homologous chromosomes move toward opposite poles of the cell. Chromatids do not separate. Homologous chromosomes move towards the opposite poles of the cell. Chromatids do not separate. Chromosomes gather together at the two poles of the cell and nuclear membranes reform. Homologous chromosomes pair and exchange segments, right? Then homologous chromosomes align on a central plate. The haploid cells separate completely. Now, this is regarding the cell division, means this is regarding the meiosis process. You have to understand, because there it is given that the homologous chromosomes pair and exchange segments, no? So the pair and exchange segments means that is meiosis. Got it? That is the crossing over, right? It is not like the crossing of the Devinini and the Sangita, right? It's the crossing over taken place in the what? It is in the cell division. It is a meiosis, right? In the prophase 1. So actually the first one is homologous chromosomes pair and exchange segments. That is the first one. So the C has to be the first one. Then homologous, it is happening in the prophase 1, no? Right? So of the statement in the fifth one, you have to separately name them. Now get touching with the things we are discussing in the class, right? In our the revision, I'm always focusing on the MCQ point, and the video I have done in explaining the point analyzing of the MCQs and the SCQs which were tested in the A-level, that they are giving some certain particular questions which will be frequently asked that are known as the common MCQs, right? Common MCQs with the common pattern, right? So such kind of a thing is the fifth one. So the cell cycle or the cell division is always tested. Now in the fifth one, C is the first one, and it is taken place at the prophase 1, right? Then the other one is the D. Homologous chromosomes align in the central plate. It is happening at the metaphase 1. It is happening at the metaphase 1. Then the other one is the A. The homologous chromosome move toward opposite poles of the cell. Chromatids do not separate. So move towards the opposite cells means that is the anaphase 1. Anaphase 1, right? Then next one is the B. Chromosomes gather together at the two poles of the cell and nuclear membrane reform. It is the telophase 1. Then the final one is the E. The haploid cells separate completely. The haploid cells separate completely. It is in the what? Cytoplasmic division, no? Right? Cytokinesis, right? Cytokinesis. It's the last one, right? This is the order. So the answer is correctly given, and it is the second one. Sixth one. Which phase of nuclear division represented? You can clearly watch that here, right? Here. It is given like that. Now, this is an animal cell. Now, the aster and spindle fibers and all those things are present, right? Here also same. right? Spindle and aster and all those things are present. And actually here comes, but now they are given like two chromatids are here, right? Here one and here two. Third one, here one, another one, and here another one, right? Right. Here also the same thing. Now, two chromatids you can separately identify, right? Two chromatids are seen, right? Okay. Then the other one also, two chromatids are separately can be seen, right? Now, this is the scenario. Now, this can be happen either prior to the metaphase, right? It means actually definitely you can understand this is meiosis, right? Therefore, no any doubt regarding this is mitosis or meiosis. This is meiosis, right? No issue, right? Regarding the meiosis, what happened? It has to be definitely it has to be prior to the prophase, and then it is prior to the metaphase, so either after the metaphase. So prior to the metaphase, it has to be the prophase one. Are there any answer like that prophase one? No. Actually, it has to be the meiosis. Therefore, you can just cut down the answer 1 and the 4. Then 2, 3, 5 separate. Then just after the metaphase means it has to be the what? It has to be the anaphase, right? It has to be the anaphase, right? Definitely. Then it has to be anaphase. Now, this is taken place now in the middle, right? In the middle, that is meta, right? Here, what happens now? Here, the metaphase is there, middle, right? Then prior to the metaphase and after the metaphase, right? Prior to the metaphase, what is that? Here, it has the prophase, right? Pro. Then after the metaphase, it has ana. It means ana 1 and it means meta 1, ana 1 and pro 1, right? So prophase 1 is not given. So therefore, we can clearly get the answer as anaphase 1. Therefore, the answer for the sixth one is anaphase 1 of meiosis, right? Answer for the sixth one is fifth one. Seventh, which of the following cellular organelle causing cell death by autolysis? That is the lysozyme. It is a just straightforward theory fact. Therefore, the answer for the seventh one is second one, right? Select the correct statement with regard to the C4 mechanism of photosynthesis, right? C4 mechanism of photosynthesis, right? It is also very easy. Now we can take it like this. Now the C4 photosynthesis is taken place in two different cells, right? In the outer, right? It is taken place in the mesophyll cell, right? It means externally it is now, if we take this as the cells, now I am just denoting the uppermost cell as the exterior one, then the interior one as the lower cells, right? There are now two cells. Here the plasmodesmata are present to separate the cells. Likewise, right? Okay. Right, this is the thing. Now what happened? Here, first carbon dioxide gas is here and it is coming to the cytoplasm now. This cell is the what? This is the mesophyll cell, right? This is the mesophyll cell, right? Then interior, it is the bundle sheath cells, right? Bundle sheath cells. Bundle sheath cell is interior, right? Then carbon dioxide dissolved in water and it forms bicarbonate here. Then this bicarbonate along with PEP, right? Along with PEP and binds to the PEP and they form, what is they are forming? They are forming the oxaloacetate, right? Oxaloacetate. Now, this is the process now. Then oxaloacetate turn into, it is stable, but it is turn into malate to aspartate, which are more stable than the oxaloacetate, right? Malate to aspartate. Then this malate to aspartate goes through the plasmodesmata. Then I will take the malate as the component. Then they just diffuse to the bundle sheath cells. Then there what happened? In the bundle sheath cells, malate is converted into the pyruvate, right? Malate is converted into pyruvate, and this pyruvate just again goes through the plasmodesmata again back into the mesophyll cells. Then pyruvate is turned into the PEP. This is the process, no? Now there's a critical thing happening. From the scenario to pyruvate to PEP, that ATP one is needed, right? So it produces ADP then and inorganic phosphate, right? So ATP is used here, right? The energy is used. And here also another thing happens. Then oxaloacetate is converted into malate. What happens? The NADPH is the energy in here is used and they just oxidize into NADP plus. These are the two aspects here. Then there's another important one. During the conversion of malate into pyruvate, carbon dioxide is evolving, right? Evolving of carbon dioxide. Then what happened? Inside the bundle sheet cells, this carbon dioxide is grabbed by the RUBP. Then it lean into the cycle of formation of what? Calvin cycle. Calvin cycle take place, right? Now this is the aspect, right? Okay, now Calvin cycle can be found in the bundle sheet cells. Now PP involvement of the it means by the bicarbonate bind into the PP that forms oxaloacetate. There is also carbon fixation. Here is also carbon fixation. Now here bicarbonate fixation and here the carbon dioxide fixation. There are two carbon dioxide fixers here. In the mesophyll cells, that is the PP. Now we'll go through the carbon numbers of these things. Then PP having 3 carbon, right? PP is 3 carbon. Then bicarbonate 1 carbon. Then oxaloacetate means 4 carbon. Then again malate and aspartate means 4 carbon, right? Here again 4 carbon. Then pyruvate carbon is just evolving. Then pyruvate means 3 carbon. Again 3 carbon. Then again form into 3 carbon. Now this is the process. Okay, got it? 5, right? Okay, so this is the scenario taking place. Now we'll go to the answers. First carbohydrate derivative produced is a C4 compound. Yeah, the first carbohydrate derivative product is here, no? Now this is the oxaloacetate and it is 4 carbon, right? It is correct. First carbohydrate derivative is oxaloacetate. First answer is correct. Fixation of atmospheric CO2 occurs within the cytoplasm of the leaf mesophyll. Actually, the carbon dioxide fixation is not taking place, right? It is a bicarbonate fixation because carbon dioxide entering from the atmosphere reach into the cytoplasm of the mesophyll cells. Then at the cytoplasm, you know that cytoplasm is a liquid portion, no? It's liquid portion. Now this carbon dioxide tend to just dissolve in water and form the what? The carbonic acid, H2CO3. In the chemistry, you getting to know that thing, right? Carbon dioxide react with water to form by this carbonic acid, H2CO3. Then that carbonic acid is an weak acid. In that scenario, it is break down into the H plus and bicarbonate. So this that the formed bicarbonate fixed by the PP, right? Here in the PP bicarbonate fixation, there is an enzyme. What is that? PP carboxylase, right? That particular enzyme, just right? It is the enzyme which what catalyze this particular reaction. And here also, for the fixation of carbon dioxide with the RUPP, there you have the Rubisco enzyme, which catalyze the fixation of carbon dioxide with the RUBP. So those two enzymes are used here, right? And also in the carbon dioxide turning into bicarbonate, it means carbon dioxide dissolving with water, then carbon dioxide turning into bicarbonate, it is what? It is catalyzed by the reaction, by the enzyme carbonic anhydrase, right? Carbonic anhydrase. Those are the crucial points, right? Okay. So that is the point. So carbon dioxide is, so the bicarbonate is catalyzed by the carbonic anhydrase. Now second one is incorrect. Therefore, carbon dioxide fixation, no, that is the bicarbonate, right? Third one, photorespiration does not occur due to the absence of Rubisco. Actually, in the C4 plants, there is no photorespiration. That is just another deep explanation. Now, tomorrow, at the session, there will be a session. Tomorrow, we are discussing about the cellular activities. There, I will explain you about the cellular activities regarding the, it means photosynthesis, right? Then we will discuss about the photorespiration and all those things, right? Okay, got it. So then malate is turned into pyruvate, right? Okay, carbon dioxide is evolving and that is fixed by the RUBP. So Rubisco enzyme is present, right? There is no absence. There are some certain reasons that why the photorespiration is not taking place in C4. There are other reasons, right? That is not because of Rubisco absence, right? Calvin cycle occurs in leaf mesophyll. No, Calvin cycle is occurring in the bundle sheath cells. 3-phosphoglycerate is not an intermediate product. No, you now get to know here the Calvin cycle is taken place. Then Calvin cycle, during the Calvin cycle, we know that the 3-PGA is formed. 3-PGA means 3-phosphoglycerate. In the mechanism of Calvin cycle, we are forming the 3-phosphoglycerate. Now here the Calvin cycle is taken place, then just indirectly suggested that there the 3-PGA formation is there. Fine. So therefore the answer for the 8th one is first one. Answer for the 8th one is first one. 9th. Which one of the following probably evolved first? This is a very, it means very creative question, right? So it is asking about which one of the following probably evolved. Actually, you get to know there is a point. It was tested in the 2020 MCQ actually with the A level that I faced, right? With the MCQ paper, first MCQ of mine A level paper, right? 2020, right? So they ask about who are the first organisms involved. They are the anaerobic prokaryotes, right? Right? Anaerobic heterotrophic prokaryotes. So therefore, then there it gives a clue. Anaerobic means they just do the anaerobic respiration. So anaerobic respiration, we know now, right now the anaerobic respiration, it is independent of the oxygen. It means it is not needed, that oxygen is not needed, right? Whether oxygen present or absent, the glycolysis taken place because of the anaerobic respiration pathway. So then you have to know now the Krebs cycle, it is come under aerobic. Then electron transport chain, it is also comes under aerobic. Then splitting of water comes under photosynthesis. Now the photosynthetic organisms were evolved just after all those things, right? Okay. The cyanobacterians were the first photosynthetic organisms to evolve, no? That is just after the long period after the this process, right? So after the those things, right? Then Calvin cycle is also comes under photosynthesis. Now, prior to those photosynthesis and aerobic respiration, what was happened? The scenario happened was the anaerobic respiration, and the glycolysis is there, right? The glycolysis is there, so therefore the probably evolved first is the glycolysis. Answer for the 9th one is first one. That is a creative one, right? It is not a theory point which is given in the resource book, but you have to argue with yourself and derive an answer, right? So 10th one. The amphibians dominated in the period of Paleozoic. That is an easy one. One for the 10th one, right? Now better to tell how much out of 10 you have taken for the first 10. How much out of 10? Quickly tell. How much out of 10 you have obtained for the 10 questions, right? Very good. We'll move to the last 10, right? We'll move to the last 10 from the 41 onwards for the next discussion, next part, right? Right. 41st question. Come to the 41st. Which one of the following statements or the statements regarding enzymes is correct? pH alters chemical bonds involved in the formation of enzyme substrate complex. Actually, the pH can alter, but you can't say that is pH, no? That is the pH which is apart from the optimum pH, no? Therefore, the certain particular enzyme reaction to take place, we need the optimum pH range. So it has to be a kind of a extreme pH condition. It may be just less than the optimum pH range or higher than the pH range. So you can't say that certain particular pH alters the bonds, right? So that cannot be taken as correct. Okay, just understand the point there, right? Okay, therefore A is incorrect. A is incorrect. We can't directly say that pH alters. It has for that statement to be correct, it need to be like this. The pH just deviating from the optimum pH range can alter the chemical bonds, right? Okay, right. The second one, some membrane proteins act as enzymes. That is accepted, right? some proteins found in the plasma membrane, they can act as enzymes. In some enzymes, beyond the optimum temperature, active sites do not change. You can't say so. Each and every enzyme, they just fall down when their optimum temperature is deviating, right? So for the human enzyme, it can be 35 to 40, right? Likewise, so if it is an archibacteria, then it can be 80 to 100, like that kind of a range. But even for the archibacterians, when the temperature is more than 100, then their enzymes also get denaturated, right? Their protein also get denaturated, then the enzyme also fallen down, right? Okay, there is no catalytic reaction then after. So you can't say that in some enzyme, not each and every enzyme, but the optimum temperature might be varied according to the human enzyme, or even in our body also. If we take 20 enzymes, then 20 enzymes might have 20 different optimum temperature values. But that value it is in between the 35 to 40. Then in archeans also, it is between 80 to 100, right? Okay, got it. So therefore, it is incorrect. D. Allosteric activator always bind to the active site, right? Okay, it can't wait. Allosteric activator always bind to not the active site, they bind to the regulator site. Therefore, the D is incorrect. They are not either the allosteric activator or the allosteric inhibitor. They are not binding to the active site. They are binding to the regulator site, right? We have discussed those points in the CT, the earlier CT which were held in the in the Friday, you know, right? Actually, that was a YouTube live stream, so anyone can go to that video and check whether those points, right? The CT session of the critical thinking is actually the heart of our paper class, right? It is the heart of the paper class. There we discussed the guessing MCQ point of this year, as well as the common MCQ points, right? Which are always asking in the A-level exam. So that CT session is massively very helpful for each and every student to increase their marks, right? So don't miss that, right? Okay. Then E, allosteric inhibitors are irreversibly non-competitive inhibitors. They are non-competitive and they are not irreversible, they are reversible. Therefore, the E1 is also incorrect. So the answer for the 41 is 5. Answer is only B. Only B, right? Okay. Then 42nd. 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 Select the correct stress response adaptation in plants. Increasing the proportion of unsaturated fatty acid in membranes by cold stress plant. Now what happened? When the cold stress is increasing, it is actually directly proportional to the solidity of phospholipids, right? In order to now the solidity of the phospholipids, right? So then what happened? The stability of the plasma membrane, that is inversely proportional to the solidity of phospholipids, right? So therefore, in order to bring back the stability, we need to decrease the solidity, no? Now, solidity of phospholipids, it is directly proportional to the saturated fatty acid content, right? It is directly proportional to the saturated fatty acid content. So in order, we have to decrease the solidity of the phospholipid, we have to decrease the saturated fatty acid content. It means saturated fatty acid content. In the other word, you can say now solidity of the phospholipid is inversely proportional to the unsaturated fatty acid content, right? Got it? So then so we can say that the stability of plasma membrane directly proportional to the unsaturated fatty acid content, right? These are the MCQ points, right? Okay. Increasing the proportion of unsaturated fatty acid in membranes by cold state plants. That is correct. So in order to maintain the stability, we need to increase this unsaturated fatty acid content. That is correct. B. Decrease in cytoplasmic sugar levels before winter in frost tolerant plants. Right? What happened in the frost tolerant plants means it is also in the cold states, right? Now you can better take a screenshot or you can quickly write down those things. In the cold states, what happened? The things get melted, right? Okay, they try to get frozen. Then the cytoplasm and those water content, they just tend to freezen, right? In order to decrease that, the solute potential or the solute concentration of the cell need to be increased. What? To avoid the freezing or minimize the effect or the adverse effect caused by the freezing, we need to increase the solute concentration. So prior to some cold season or frozen season, what happens? Plants just increase the solute concentration of their cytoplasmic contents, right? In a cold stress, what is happening? They just increase the solute concentration of their cytoplasmic levels. Then once the solute concentration increases, that the freezing getting decreased, right? I will write that also. Now I think you have noted down these things. If you miss that, you can take a screenshot, right? Okay. Right? What happened? Solute concentration of cytoplasm. Solute concentration of cytoplasm actually increases with the cold stress. Now this is what happening. In the cold season, the solute concentration of the cytoplasm is increased. When the cold stress is increasing, that the solute concentration of the cytoplasm also increases, right? So decrease in the sugar levels before winter in frost-tolerant plants, that is incorrect. C. Presence of organic compounds in cell sap of salt-tolerant plants. Now what happened? Normally now this is happening. No? Now if this is a root, now this is the soil solution. Now imagine this is the soil solution. Now the plant absorbing water molecules into the root. No. What is the reason? The reason is here inside the root, if we name it as psi W root, then here psi W, not psi W, psi root, no need of a W, right? Psi root. Then we here name it as psi soil. Normally, what is the condition, right? Normally, the condition is psi soil is far more than the psi root, right? It's higher than the psi root. It means water potential of the root. Okay. So then water tends to go inside the root. But in salt conditions, what is happening? In salt condition, it means in salty water, what happened? In salty water, psi is increasing. So that psi S is increasing, that tending to the decreasing of the psi. Decreasing of the psi. So therefore, what happened? That the water potential gradient seen here. It means the water potential gradient of the psi soil and the psi root. What? Water potential gradient between the xy soil and the xy root is decreasing, right? It's decreasing. So that tend into the there might be a problem in the transportation of water into the root hairs, right? Root cells. Okay, got it, right? So then what happened? To overcome that, now the size of the soil solution is also decreasing. No, then what that happened? What that leads into? It leads into the size decrease in the root also, right? It means that is what happen inside the plant. That is adaptation of the plant. To that case, to decrease the psi root, what they are doing, they are increasing the psi s of the root. They are increasing the psi s of the root, right? So in order to that, they are increasing the concentration of the root cells, right? They are increasing the concentration of the root cells. Presence of organic compounds in cell sap of salt tolerant plants. That is correct. D. Release of abscisic acid by drought tolerant plants. That is also correct. E. Secretion of excess salts from leaves by salt tolerant plants. Yeah. In the salt tolerant plant, if we take this as a salt tolerant plant, then they have leaves like this, right? Then what happen in there? They are living in such environment where the salt concentration is high, right? In their soil solution, the salt concentration is high. Therefore, with the water intake, they are taking more salt inside to the plant. That is not favorable for the plant. So what happened? The plant need to remove the excess salt out from the plant. For that certain particular thing, they just develop salt glands in their leaves, right? That is also correct. Then correct answers are, so the answer is 5 because the answers are A, C, D, and E. Right? Okay. Then we'll go to the 43rd one. Common feature or features to all vascular plants is right? Heterospore is not found in all vascular plants. There are homosporous plants like Nephrolepis, Lycopodium. That is incorrect. Branched vein leaves are only branched vein leaves, no? Right? No, that is not also found in all vascular plants. The plants which are, it means they are the dicot plants have the reticulate venation, no? The monocots have a parallel venation which is unbranched. Therefore, it is incorrect. Dominant sporophyte, yeah. Dominant sporophyte is found in all vascular plants. That is a crucial point. All vascular plants have a dominant sporophyte. All the non-vascular plants have a dominant gametophyte. That is a very crucial point. All the what's the point? All the vascular plants have a dominant sporophyte. All the non-vascular plants have a dominant gametophyte. Okay, got it? Got it? Write down that also. Therefore, for the 43rd, A is incorrect, B is incorrect, C is correct. Presence of embryonic state. That is also correct. There is no embryonic state in non-vascular plants, right? Wait. It is not talking about non-vascular. No, right. Presence of embryonic state is found in all vascular. No need to just go and compare with the non-vascular, right? Xylem vessels are not found in all vascular plants. Xylem vessel is found in all the angiosperms and also Nitophyta, right? Nitophyta and the angiosperms. It means the flowering plants and Nitophyta have the xylem vessel element. Others don't. So therefore, the answer for the 43rd is 4. CD. Forty-fourth. Which of the following are the functions of connective tissues? Protection, transport of materials, not secretion, insulation, no absorption, right? Answer is, for the 44, that is very easy question. Answer is 1. Answer are A, B and D. 45th. Which of the following digestive enzymes or enzymes produce end products that are absorbed directly in the small intestine, right? Which of the following digestive enzyme or enzymes produces end products that are absorbed directly in the small intestine? We'll see. Now this is a very crucial point, right? Now this is a very crucial point, right? Now you know, right? Small peptides. Then what else? Amino acids. Then glucose. Then vitamins. Okay. These are the substances which are absorbed, right? According to the question, absorbed directly directly in the small intestine. Now write down those things. Small peptides, amino acids, be quicker, right? Glucose and vitamins are absorbed directly in the small intestine, right? They are absorbed directly in the small intestine. These are absorbed directly in the small intestine, right? Okay. Now you have to go through the enzymes and check whether what are the end products, what are the end products, right? Okay. Now first one, salivary amylase, right? Now salivary amylase, what it is done, right? Starch is converted into, right, small polysaccharides and disaccharides, right? Okay. Then pepsin, what it does, it converts protein into small what? Small polypeptides. Then chymotrypsin converts small polypeptides into smaller polypeptides, right? Then carboxypeptidase converts smaller polypeptides into amino acids, right? Then pancreatic nuclease converts DNA and RNA into nucleotides. Now what is the enzyme here? Salivary amylase. Now what is the enzyme here? Pepsin. What is the enzyme here? Chymotrypsin. What is the enzyme here? Carboxypeptidase. Then what is the enzyme here? Pancreatic nucleus. Now, you can see here, these are not the directly absorbed components in the small intestine. This is also not. This is also no. Yeah, this is okay. And nucleotides, it is not mentioned, right? Actually, it has to be the sugar type, no, the ribose or deoxyribose sugars, and then the phosphate, and those are the directly absorbed, not the nucleotides, right? The nucleotides further has to be broken down. So therefore, only the answer is what? Carboxypeptidase, only D. Answer for the 45th one is D only. Answer is 5. Reason is D only. This is a very good question, right? 45th one. This is an excellent question. So you need to have so many things around in your brain, in order to answer this. First, you have to go through these enzymatic reactions also. What is the substrate? What is the end product? And what is the enzyme? It is catalyzing. And also then you have to have the knowledge of the memory regarding the direct end products of the absorption process in the small intestine. So only you have those two. It means you definitely need to have those two. The students who are having both those things in the memory can only answer the question, right? Okay. 46. Which of the following animal nitrogenous excretory organ, right, combinations is and are correct? Yeah. Okay. Prawn has green glands, done. Correct. Cockroach having Malpighian tubules, correct. It's an arthropod, no? Spider has book lungs. That is also correct. Spider and the arachnids having the book lungs, no? Right? Marine reptiles having salt glands. That is also correct. No, actually it is asking the nitrogenous excretory organ, right? Oh, that is tricky. That is very tricky, right? C is correct actually. Spider, book lungs, but it is asking the excretory product, right? Hey, don't be a fool, right? Myself also just get trapped into my own trap, right? Okay. Book lungs is a respiratory structure. It is not a excretory structure, right? Okay. Then also, I think if someone has taken C as correct, then you have to just have a shot at your head, right? Then marine reptiles, salt glands, okay. Human sebaceous gland, no, that is the sweat gland. So answer for the 46 is 1 ABD. It's better you tell in the chat those who have fallen into the trap of the book lungs, right? Yeah. You have noticed prior to me, no? Very good. It's given in the, yeah, stated in the chat, right? Forty-seventh one. Yeah, okay, yeah. Actually sweat glands is a part, right? The main excretory one is asking, no? Main. Oh, actually, that is organ it is asking. So when asking about the organ, it has to be the kidney, right? We can take the sweat glands also, but that is not an organ, no, that's a structure. Then organ aspect, right? Dilani, I think you will get that, right? Dilani is asking that. The kidney is the organ aspect. If it is asked about the structure aspect, we can take the sweat glands also. Fine, right? 47. Homozygous dominant pea plant having yellow round seeds was crossed with homozygous recessive pea plants having green colored winkle seeds. Dominant is the yellow and round seeds, no? Yellow will put capital Y, capital Y. Homozygous dominant, no, round we will put capital R, right? So yellow means capital Y, round means capital R. Then it is crossed with homozygous recessive pea plant having green color wrinkle seed. Green will take simple Y, then wrinkle will take simple R. This is a cross, no? If the alleles show independent assortment, which of the following statement is and are correct regarding the F1 progeny of the above cross? Then they are showing independent assortment means we can take the normal crossing, normal dihybrid crossing, right? So they get into the YR for the first cross. They the gametes, then simple Y, simple R the gametes. Then this gamete fuses, right? It forms capital Y, simple Y, capital R, simple R in the F1 progeny. It is asking about the F1 progeny, no? F1 produce gametes with 4 genotypes. Gametes with 4 genotypes. Okay. Yeah, F1 produce gametes with 4 genotypes. No, this is only a one single genotype, no? So the F1 progeny is 100% yellow and round, no? That is incorrect. 4 phenotypic groups would obtain by self-pollination of F1 plants. Yeah, it can be. 4 phenotypic groups would be obtained by self-pollination of F1. This is a normal dihybrid cross. You know, now if Y, capital Y, simple Y, capital R, simple R cross with capital Y, simple R, capital R, simple R, will take the ratio, will get the ratio at 9:3:3:1. This is a phenotypic ratio. So we have 1, 2, 3, 4 types of phenotypes, right? That is correct. Then B is correct for the 48, right? For the 48, A is incorrect, B is correct. When F1 subject to a test cross, 50% of the plant progeny possess dominant genes for the both characters, right? 50% of the plants. Then F1 is subject to a test cross. Test cross means capital Y, simple Y, capital R, simple R into simple Y, simple Y, simple R, simple R. We'll see. Then here what comes simple Y, simple R. Here the gametes are capital Y, capital R, capital Y, simple R, simple Y, capital R, simple Y, simple R. These are the gametes, right? Okay. Right. Genetics is very easy. Then what is the progeny coming? Capital Y, simple Y, capital R, simple R2. Capital Y, simple Y, simple R, simple R2. Simple Y, simple Y, capital R, simple R2. Simple Y, simple Y, simple R, simple R. Right. 50% of plant progeny possess dominant genes for both. Dominant genes for both with, right? Okay, only this one has the dominant genes for both. Only this, no? So as a percentage, that is 25%, no? So that is incorrect. C is incorrect. D. 8 genotypic groups would be present in the progeny obtained from self-pollination of F1. Yeah. Then what is the, when we go to the this cross, now genotypic aspect, we'll get the genotypic amount from 2 to the power 3, no? Right? 3 is the index we use, 2 because this is a dihybrid cross. This is dihybrid. Therefore, 2 to the power 3 means 8 different types of phenotypes. Sorry, genotypes. That is correct. Genotypic ratio can be obtained like that. And also for the phenotypic ratio, we can take the 1, 2 to the power 2. Right? The 2 because this is a dihybrid cross. Here also the same. Here also the same. For the genotypes, we use the 3 as the index. Then for the phenotype, we use the 2 as the index. That is normal in all genetics, right? It's kind of a short method, right? Okay. Then C, D is correct. E. Ratio of homozygous dominant plant obtained from self-pollination of F1 is 4 out of 16. Homozygous dominant plant obtained. Yes, we need to make the Punnett, right? Okay. We need to make the Punnett. I think it will be a time wasting one. We have to complete the Punnett square for this one. Actually, we'll get as 4 as the genotypic ratio. 4 out of 16. That is also correct, right? Is also correct. You can better to make the Punnett chart for this cross. Then you will take the. It means it is asking about homozygous dominant plant obtained. No, homozygous dominant means, right? Homozygous. This ratio they are asking. Go to the Punnett chart and you will take get 4 of this, right? That is correct. Right. Therefore, answer for the 48 is 5. Answers are B, D, E. Right. Okay. 49. Which of the following statement is correct regarding sterilization? Microorganisms are killed by oxidation in the hot air sterilization. That is correct. Pasteurized milk by ultra high temperature can be stored for several months without refrigeration. That is correct. As UV radiation is truly penetrating, they are non-penetrating, right? Then UV radiation doesn't normally penetrate to subject, right? Okay, got it. Moist heat sterilization is commonly used to sterilize temperate labile fluids. That is also correct. Endospores of bacteria can be destroyed, right? Using the chlorine dioxide method of chemical sterilization, we can just destroy the endospores, right? Okay, of some bacteria. Therefore, for the question number 49, answer is 1. Answers are A, B, and D. For the 49, that is the 1. 50th. Which of the following statements regarding coliform bacteria is correct? Actually, the coliform bacteria, when we regard the coliform bacteria, that is used in the process of just purification of water, right? To find out whether the water is contaminated, right? It's the kind of the detector microorganism, right? Then the coliform, we know that is aerobic or shows the anaerobic or the facultative anaerobic. Aerobic or facultative anaerobic. Then it's a gram negative one. Then non-endosperm forming. It means no endosperms are formed by the coliform. Then it is rod shaped, right? So they are, then we'll go through the question. They are non-pathogenic. Yeah, they are non-pathogenic. They are found in the lining of the intestine of the human alimentary canal. That is correct. And they are non-pathogenic. If we have coliform in our intestine, no issue. That is not pathogenic. That is correct. They form endospores. That is incorrect, right? They are not forming. They are indicator organisms to detect fecal contamination in water. Yeah. Because that those coliforma live inside our intestine. Once our feces are passed away, if that the water is contaminated with the feces, right? Then we can detect if the water, if we find coliform in the water, because normally the coliform is found in the intestine and all these plants and those things, no, it is normally not found in the water. So we can detect if the coliform is found in a certain water solution or the water. If we take a water extraction, if we found coliform there, then we can detect that water is contaminated by the feces, because our feces contain coliform, because that the coliform is found in our intestine, right? Okay, that is also correct. It is correct. Some coliform found in plant and soil samples also, that is also correct. They are not gram positive, they are gram negative. Therefore, answer for the 50th one is 2. Answers are A, C, and D. Correct ones are A, C, and D. Right. Better to right. Okay. I missed the 48th. Okay. Pardon. Right. I will tell it also. Right. Okay. 50th answer is 2nd. I have missed the 48th one. Right. Okay. I will tell the 48th. There are no trees in the savanna grassland. Actually, the characteristic feature of the savanna is the what? The scattered trees in the tall grass cover. No? Scattered trees. Here and there can be scattered trees found in the savanna. So, savanna contains trees. That is incorrect. Deserts are present only in tropical region. No, they are found in both temperate and as well as in the tropical region. Right? Therefore, it is incorrect. Many trees in the northern coniferous forest have needle-like leaves. That is correct. Chaparral composed of dwarf trees with fire-resistant roots. That is correct. Tundra covers the expansive areas of not the Antarctic Circle, the Arctic Circle. Therefore, the answer for the 48 is 48. Answer for the 48 is, right? 48 is 4. Answers, correct answers are C and D. Right. Now, as good students, right, I think you have obtained good marks for the last 10. That is somewhat tricky and somewhat not easy actually. There is some sort of, there is an average difficulty in those final 10 questions. Now, better to mention your marks out of 10 in the live chat, right? Yeah, English, right? Okay, that is given that the penetration one, they are not penetrating through. Actually, they are the UV is penetrating, but it is asking about here regarding the hospital operating theaters, no, right? Mattresses, they are not piercing through the mattresses. That's the reason, right? Actually they can penetrate, right? But they are not penetrating through the mattresses. That is the point, right? Don't mess up with that. UV radiations are not penetrating. They are penetrating, but not penetrate through the mattresses. That is the point. Okay, Another, is it fine? Okay, I think it is fine. Right. We'll come from the 11th one. No, mattresses are sterilized not by the UV radiations, right? That UV radiation is used to sterilize the environment of the certain particular walls, not the mattresses. They are just boiled in the hot temperature and those are put in inside the hot water and those are sterilized in such a manner, not the mattresses, right? Okay, right? Got it? For the sterilization of the environment of the wall, for the hospital is used the UV radiation. Okay, got it? 11th. Teach a common for two domains of organisms. Presence of peptidoglycan in cell wall only found in one domain. It is the bacteria. Beginning of protein synthesis with 4-methionine, that is also only found in the bacteria. Presence of branch and chain lipids in cell membrane, that is also only found in one domain, that is the archaea. Presence of several kind of RNA polymerase enzymes is found in both archaea and eukarya. So therefore, fourth one is correct. Cellular organization is eukaryotics, that is also only found in what? Eukarya, right? So therefore, the answer for the 11th one is 4. It is very critical. It is asking about only found in two domains. No? Animal have a protrusible pharynx and two eye spots. Which of the following character could be present in this animal? Actually, this animal is a platyhelminthes. Then two eye spot means particularly this one is a white free living platyhelminthes. So what is There they need to have cilia for the locomotion because they are free living. Answer is fifth. 13. Maybe a planaria, right? The diagram given below represents a cross-section of a stem with secondary growth. Several tissues have been labeled. This is the exact CT we completed there in the Friday session. A to H. Which one of the following indicates the tissues of the bark? Tissues of the bark means this is the vascular cambium, right? Vascular cambium. So all the tissues away from the vascular cambium are included in the bark. So here, what is the vascular cambium donated by? I think by the letter F. Here, vascular cambium is donated by the letter F, right? So therefore, A, B, C, D, E are the tissues of the bark. Therefore, the answer is fourth one. 14. What is the unique feature found only in the seed-bearing plants of kingdom Plantae? That is the pollen grains. 15. Which of the following characteristics could be seen in both Nephrolepis and Selaginella? Transportation through xylem and phloem because they are vascular plants. Therefore, answer is third. 16. Which of the following could be considered as an active process, right? Transport of sugar from CO tube element of the phloem into the zinc, right? That is through the transport stomata, that is diffuse. Therefore, that is an inactive or the passive process. Accumulation of clay plus in the guard cells from neighboring epidermal cells in the process of stomatal opening, that is an active process. Second one is the answer. The transport of sugar from one CO tube element to the adjacent CO tube element, that is taken place through the bulk flow, that is a passive. Guttation takes place through the hydrothode due to the root pressure, that is also a passive. Water and solute move along the apoplastic route, that is also a passive. So therefore, the answer is second one for the 16. 17. Which of the following event would enhance water uptake by a plant cell? This is somewhat critical, right? This is somewhat critical. Now, water uptake by a plant cell means that it is regarding the neighboring cell, right? Now, there are two cells. Now, this is the cell we are considering. Now, there is another cell which is neighboring, right? Or cello may be a solution, something which is neighboring to the cell, right? Which is neighboring to the cell. Now, this is the cell we are talking. Decrease in the size of the surrounding solution. If the size of the surrounding solution is decreasing, what happens? Now, water uptake means psi here. Psi, we will take psi of the psi out. Now, this psi in, we'll say that wise. So, psi out need to be, this is a simple theory. Psi out need to be higher than the psi in. Therefore, then only the water will flow into the cell. That is the water uptake aspect, no? Right. Then decrease in the size of the surrounding solution, that is incorrect. First one is incorrect. It has to be increased. The increase in the positive pressure on the surrounding solution, right? Then psi is affected by what? Psi is affected by psi p. Now, surrounding solution, it is given maybe a cell, no? The cytoplasm of the cell is also a solution, right? You can't say this is a solution, it is put in a vessel or something. Container, it can be a solution in the cell, cytoplasm solution. So when the psi p is increasing, psi also increase. So psi increase means psi out increase. Therefore, the second one is correct. Third one, the loss of solutes from the cell. Cell solutes are losing means now psi is inversely proportional to the psi s. So psi s decreasing means psi is increasing. So psi in need to be decreased. That is the which will promote the water uptake. So psi in need to be decreased, right? So throughout this scenario, psi in need to be decreased. For this to happen, psi out need to be increased. This is a simple theory, no? Therefore, third one is also incorrect. Fourth one, increase in the solute potential of the surrounding solution. Increase in the solute potential means the psi is decreasing. No, psi need to be increased. Therefore, that is also incorrect. Increase in the turgor pressure of the cell. Once the turgor pressure increases means the psi in is already increases. That is also incorrect. It has to be decreased. So the answer for the 17 is 2. This is a very good question, 17th one. Really good question, right? Really good question. Then 18. Which of the following mineral element may cause lateral transport of oxygen? It's a very easy question. Calcium is the responsible one for the lateral transportation of oxygen. Answer is 3. 19th. Select the correct statement regarding plant A. All seed plants do not need external water for fertilization. Yeah, all seed plants do not need. They just need the internal water. It means starting from the cycas, netum, then anthophyta plants, they don't need external water. First sentence is correct. Second one, all homosporous plants do not have vascular tissues. That is incorrect because Lycopodium, Nephrolepis have vascular tissues. That is incorrect. All plants that do not bear flowers are seedless. No. All plants do not bear flowers can be seed plants too, no? Cycas and all those things, coniferophytes, then cycas, pinus, netum, no? All plants with vascular tissues do not have free-living gametophytes. All plants with vascular tissues do not, no, why? The Nephrolepis have a free-living gametophyte. That heart-like thallus, heart-shaped thallus, that is free-living, no? Free-living, no? Right? So that is incorrect. There can be. All gymnosperms do not have vessels in xylem. No, except netum. This netum is a gymnosperm, netum have vessels. That is also incorrect. So the answer for the 19th is 1. 20th. 20th, some statement regarding plant growth regulators. Auxin and ethylene produce antagonistic response for leaf abscission. Right, yes, that is correct. Ethylene promote the leaf abscission while the auxin just inhibit the leaf abscission. Cytokinins and ethylene show similar response to, no, that is different regarding the leaf senses, right? All plant growth regulators are transported from the place of production to trigger responses in target cells. No, some just maybe act on the site of the same production site, right? It means regarding the plant growth substances, it means plant hormones. Some are just, it means for an example, if this is the plant, just imagine, now this is the plant. So if this is the leaf, some plant hormones are produced in the leaf and they are acting on the leaf. So therefore, they no need to be transported. But some plant hormones which are synthesized in the stem need to be transported to the leaf. So those need to be transported. Here it is saying all the plant growth regulators are transported, no, not all. So in that aspect, you need to be careful. So therefore, 20P only correct. So therefore, the answer for the 20th is also 1. Now, quickly tell me how much you have obtained from 11 to 20 out of 10. Out of 10, how much you have obtained? Right, okay. 21. Peristalsis occur. Now we'll go through. Peristalsis occur in human alimentary canal from the beginning of the esophagus to the rectum. When it is starting from the esophagus, it travel entire through the esophagus, stomach, small intestine, large intestine until the rectum that is taken place, right? So the answer for the 21 is 2. These are very simple. When the oxygen level in the blood is too low, we'll see. the kidneys release erythropoietin. Yeah, the kidneys release erythropoietin. The oxygen level in the blood is too low means, right? Too low. Now, what is the function of erythropoietin? Erythropoietin just synthesize the RBC, from the kidney, right? RBC are synthesized, that is stimulated by, or the activated, catalyzed by the erythropoietin. Not catalyzed, actually stimulated by the erythropoietin, right? Now, RBC production high means what is done? Hemoglobin level also increase. Hemoglobin level increase means the O2 transportation also increases. For the answer for the 22nd is also first one. We'll see the rest. The liver produces more bile. There is no connection between bile and the blood oxygen, right? The thymus, thymus is not producing red blood cells, right? That is incorrect. Sickle cell disease occurs because of the alteration of the red blood cells shape. That is not regarding the oxygen level in the blood is too low, right? The pancreas, actually, the sickle cell disease, other side, sickle cell disease lead into the blood level of the oxygen to become low, right? The pancreas secretes more glucagon. There is no relationship that also, right? 23rd. As blood enters the systematic capillaries, right? Okay, right. For that one, I will just share screen a simple thing that is actually given in the resource book, right? That's a simple diagram for that question. Now you can see. Now look here. Now this is the very important diagram. Actually, this is a very important point. I might think. Actually, I am suggesting this will be can be definitely can be tested in your A level too, right? Okay, right. We'll see. Now here, you can see now in the exhaled air, in the inhaled air, how much O2 is there, how much CO2 is there. You need to be keep in mind. You need to be just keep those data, and you can just argue, no? In the pulmonary veins and the systematic arteries, O2 concentration need to be higher than CO2, no? Right? In the other side, hindsided, in the systematic veins and the pulmonary arteries means they are carrying non-deoxygenated blood. So therefore, CO2 need to be high. So in the body tissues, you know that CO2 is high, O2 is less. Therefore, that is why the O2 is given to the body tissues now. But you need also get the knowledge that in the alveolar spaces, O2 concentration is higher than the CO2, right? So in the inhaled air, you know that O2 concentration is higher than the CO2, and also the other point, in the exhaled air also, the O2 concentration is the highest because inside there is no much CO2 more than the O2, then that will lead into the acid increase and acidosis conditions, right? So this is the chart. This is what it is tested. PO2 in the blood is same as the PCO2 in the tissue. PO2 in the blood is can be 104 or maybe 40, right? So we can't say always, right? PO2 in the blood is same as the PCO2. PCO2 in the tissue is just less than the 45. No? It's greater than, sorry, it is greater than 45. So in the blood is, it is 40 or maybe 45. So it is greater than 45. Therefore, the first one is incorrect. PCO2 in the blood is lower than that in the alveolar. PCO2 in blood is lower than that in the alveolar. Alveolar, what is the PCO2? Right? Alveolar, it is the PCO2 is 40. In the blood, what is PCO2? It is 40. It is equal, no? It is equal and it is 45. So PCO2 in the blood is not lower. It is equal, no? It is higher than the alveolar air, no? It is in the alveoli, that is 40. So in the blood, it can be either 45 or 40. It is equal or greater than. Right? Okay. Then PO2 and CO2 in the blood are in equilibrium with those of alveolar air. The PO2 and CO2 in the blood are in equilibrium with that in the That is equal, no? In equilibrium. No, PO2 in the alveoli is also 104. PCO2 in the alveoli is also 40. In both blood and the alveoli. That is correct. Third one is correct, no? That is equal, no? Same value, no? For the second one and the third one. Then PO2 in the blood is higher than that in the alveoli. Yeah. PO2 is higher in the blood, is higher than that in the, no, that is equal, no? Equal. PCO2 in the blood is higher than that in the tissues. The systematic capillaries, no, that is what is asking here. Now we need to go through the systematic capillaries, right? Systematic capillaries is asking now here, this side, the red side is asking in the question, no? PCO2 in the blood is higher than in the tissues. PCO2 in the blood is not higher than in the tissue, no? In the body tissue, that is greater than 45. So PCO2 in the blood is only 40, that is less than, no? So therefore the fifth one is also incorrect. Answer for the 23rd is third. No, better to, in the review, better to draw this, right? This is very crucial, very crucial. You will understand the importance of this in the A level, right? I think in this time, they all, it means every time they are given a hidden point, right? Hidden point, but it is very important. I think in 2023, this point will be tested, right? I will discuss it furthermore in the, Yakuteka bio session, and that session also, and in the revision, we have done those things, right? General revision, right? Okay, those things, right? Okay, I think you have got the knowledge. Okay, drop me a message to the chat, those who have understand a certain particular question. Therefore, the answer for the 23rd is 3. We are taking that from that particular graph, right? Just drop a message to the chat, live chat. Did you understand that? There is a question asking, so people living in high altitude and experiencing physical stress get low O2 concentration in blood. So it induces biconcave disc cells to sickle shape cells, right? So isn't fourth statement correct? Right? Yeah, actually we can take the fourth one. Yeah, wait. Oxygen level in the blood is too low. Yeah, in the high altitude people, the blood level, oxygen in the blood is too low, no, right? Okay. Okay, in the hindsight, that can be affect, right? Because of the sickle cell disease, both sides can be taken, no? Oxygen blood level decrease because the shape is that altering, no? Therefore, the oxygen transportation limited. In the other side also, when the oxygen amount is low, also it lead into the, right? Okay, that is also correct, right? So we can take the fourth also for the 22nd. Very good, huh? It's a good point. It is asked in the live chat, right? Very good. Okay. Other thing also, okay, no? 23rd. 25th. In man, the functional residual capacity of the volume air is the remain in the lungs at the end of a tidal expiration. That is a definition-wise question, right? Fifth one is the answer. 25th. Which of the following is the most possible pathway for the flow of blood through the human heart? Right? From the aorta, inferior vena cava, pulmonary artery, pulmonary vein. That is correct, no? Right? I will just roughly draw the chart very roughly, right? What is happening here? Now this is now this is the heart. We'll take this as the heart. Now it has tricuspid in the right and bicuspid in the left, and it is separated like that. Here pulmonary artery, here pulmonary veins and all those arteries and veins, inferior, and those are draining in. Now what happened? Now aorta runs through here, and it comes back here with superior and inferior vena cava, no? Okay. It comes with superior and inferior vena cava from the here. Then what happened? This blood goes back again into the right lungs, and it reaches from the pulmonary veins, goes back from the pulmonary artery, then pulmonary veins. What is happening? Now here from the aorta, blood which is going here, now will go through the pathway, right? Now this is the aorta, it is going through the aorta. We'll imagine the blood is coming from the inferior part of the body. Then the blood will come from the inferior vena cava, no? From the inferior vena cava, it drains into the right atrium, then back through the tricuspid, they drain into the they drain into the right ventricle. Then through the right ventricle, they drain into the pulmonary artery, and they go to the lungs. And from the lungs, they are coming back through the pulmonary veins, and they reach to the left atrium. Then through the bicuspid valves, they reach to the right atrium. This is the pathway. No? So the answer is correct given in the third one. 26. What is the blood group of a person only having Rh antigen? We have discussed those things in the CT session, right? Having Rh antigen and antibody B. Antibody B means inside they have B antibody. It means they have what? A in the in the plasma membrane as the agglutinogen. Then Rh found means positive. So it has to be A positive. Answer is 2 for the 26. This is a very easy question in on behalf of what we have tested all so far, right? 26. In human, the first line of defense against pathogen include the presence of acid in the stomach. It comes under first line of defense. It means that is the it is the normal immunity which is found in the animals, right? Okay. It is not the acquired. It is the innate immunity, no? 27 answer is 2. All the others are acquired and adaptive immunity. 1, 3, 4, and 5 comes under the acquired immunity, right? Okay. 28. During ultrafiltration in the normal human kidney, which was the following passes into the filtrate? That is the glucose, because those albumin, leukocyte, platelets, and erythrocytes, they are much larger than the pore size of the glomerulus, right? Glomerulus. It means in the glomerulus, in the filtration process, they are just larger than the size of the Bowman's capsule, right? The cells in the Bowman's capsule through the epithelium of the Bowman capsule, those leukocytes, then albumin, platelets, and erythrocytes, they can't just filter in. In one of the following groups, none of the cells undergo meiotic division. Select that group, right? This is very easy. Now, regarding the human, the oogenesis and the spermatogenesis, we need to go through that part, and we have to find whether meiosis or the mitosis occur. In the left side, I will go through the the oogenesis. We'll go through the spermatogenesis first. First one is primordial germ cell, right? This is given in the resource book, right? So what is happening? That is, we'll write the process in other color, right? Then they form into spermatogonial stem cell. Then mitosis takes place and it leads into spermatogonium, right? Then spermatogonium leads into primary spermatocyte. Right? Primary spermatocyte lead into secondary spermatocyte. Right? Then secondary spermatocytes into the spermatids. Then it into the finally sperm cell. Right? Okay. This is the chart now. Now in here, first step here in the process, it is mitosis. Here also mitosis. Here also mitosis. From the primary spermatocyte to secondary spermatocyte, it is the meiosis 1. From the secondary spermatocytes to spermatid, that is the meiosis 2. Right? Okay. Then regarding the oogenesis, we can go. Primordial germ cell, it is once again the first one, primordial germ cell. Then it turns into oogonium, right? From the oogonium, it comes primary oocyte. Primary oocyte go into secondary oocyte. Secondary oocyte leads into the mature egg. Right. Then what happens here? Here mitosis. Here also mitosis. Here meiosis. This is the process. Right. Okay. Oogenesis. This is the spermatogenesis. Then this is the oogenesis. Now we'll go back to the question. Now what it's asking group of none of the cells undergo meiotic division. Answer is 5, no? Now we'll see. Oogonia undergoes mitosis. No issue. Primary spermatocytes, they are undergoing meiosis. Therefore, first one is incorrect. Secondary spermatocytes also undergoing meiosis. Therefore, second one is incorrect. Primary oocytes undergo meiosis. Therefore, third one incorrect. Germinal epithelial cells, okay. Secondary oocyte, okay. Oogonia again undergoes mitosis. Germinal epithelial cells. Sorry, here also meiosis taken place, no? From the secondary oocyte to mature egg, also meiosis taken place. Completion of the meiosis, no? Therefore, that is also fourth one is also incorrect. Then spermatids, no meiosis. Then spermatogonia, no meiosis, it is a mitosis. Then again, ovum. Ovum is the mature egg, no meiosis, right? Therefore, the answer for the 29 is 5. In 27, yes. I just rapidly go through then 3, 4, 5. Yeah, they are the histamine from the mast cells, then production of interferon engulfing phagocytic cells. It means the macrophages, no? In the 27, yeah, actually I was just somewhat fast in the process because the single medium discussion is also there after this one, right? We have to complete it quickly, but in that process, I just told that in the 27 rest are adaptive immunity. Actually, in the 3, 4, and 5, they are just the only the innate immunity, but the factor is they are not the internal defense, they are the external defense, right? Sorry, they are not the external defense, they are the internal defense. So the external defense is the first line of defense against the pathogens, right? The internal defense is not the first line, that is the second line. So both the adaptive immunity and as well as the internal defense of the innate immunity we can take as the second line of defense. Okay, got it? Got it? There was some error in just telling the answer that was given in the chat. That is very good, right? So here and there might be some just mishaps in the discussion because this is live discussion and also time is also a factor. So we are going so fast. Then some point maybe here and there maybe doom, right? Okay, no issue, right? You can just mention it in the live chat. Okay. Now 30. Which one of the facial bone of human skull in which sinuses can be seen? Sinuses can be seen in the sphenoid, right? In the ethmoid, in the maxillary, and in the frontal bone. What are the 4 bones? They are the sinuses. Sinuses are the places where the ciliary layer is found in the where means of passage of air passages, right? Passages we are allocated for the transportation of air. Actually, we will do the skeletal system with the stimulation of the using app, right? We have done it in the general revision. We'll do it again prior to the exam in the express revision also. There will be a session for the skeletal system, right? Okay, there we'll discuss. So the sphenoid, ethmoid, maxillary, and frontal bone have sinuses. Here the answer is the okay, maxillary. Then ethmoid also present, no? So the answers, there are 2 answers for the 30. 1 and the 5th are answers, right? Both 1 and 5 are correct. Right. Then tell me out of 21 to 30, out of 10, how much you have obtained? From 21 to 30, in that 10, how much you have obtained? Just tell in the live chat. Actually in the right in the 30th. It is asking about the facial bone, right? Sorry. It's asking about the facial bone. Ethmoid is a right. It's a cranial bone, right? Ethmoid is a cranial bone. Sorry. For the 30th, 5 cannot be taken. Right. Because ethmoid is a cranial bone. Sinuses are found, but it is asking about the facial, no, the bones in the facial. Maxillary means the upper jaw, no, here this bone, right? Only that, right? Okay, thank you. 31st, we'll go to the quickly, right? We'll go quickly. 31st one. In a certain species of plant, the gene for stem color assorts independently to the gene for plant height. The symbol of the alleles of the two genes are shown below. Right? Plant 1 is a dwarf and has a black stem. Right? Okay. We'll go. Yeah. Okay. I have told, no? 30th, okay. Right? 30th done. Right? I just missed the facial one, right? Okay. Then the plant 1 is dwarf and has black stem. Dwarf and black stem. Dwarf and black stem, no? Dwarf and black stem. Then plant 1. Plant 1 has a parent that is tall and red stem. Like parent කෙනෙක් ඉන්නලු tall red stem lu. Plant 2 is tall and has a black stem. Tall and has a black stem. Plant 2. Dwarf and has a red stem. Dwarf and red stem. The number of different genotypes that would be expected in the offspring of a cross between plant 1 and plant 2. Plant 1 is a dwarf and has a black stem. Right? Plant 1 has a parent that is tall and red stem. Plant 2 is tall and black, has a parent that is a dwarf and has a red stem. Okay. The number of different genotypes that would be expected in the offspring of a cross between plant 1 and plant 2. Then again, right? It is asking about these two, right? Single words are not allowed, right? Dwarf black and tall black. Then, right. The parents are actually not needed. No, we can take now dwarf and black. Dwarf means now black will is the dominant one. We'll take. It can be BBTT or this plant can be these two. No, BBTT. No, this is the genotype. Has to be the phenotype genotype, right? Then tall black means it has to be black. No, black means tall and black. Tall and black. It's dwarf and black. Then tall and black. Dwarf and black. Tall and black means BBTT or BBTT. These two can be accepted, no? So the parent is dwarf and red. Dwarf and red means the parent has red is the now parent is simple BB, no? So dwarf means actually it has to be TT. Now BBTT. BBTT. So the parent can give only simple B and simple T. This is very critical, right? Parent can give only simple B and simple T, you know. So therefore, this one cannot be taken place because capital B is not here. It means either mother will give capital B, one parent will be capital B, but this parent can only give simple B and only simple T, you know, right? Therefore, this cannot be. Then tall and red. Tall and red means it has to be. Tall is capital T, red means, so it parent definitely be this one, no? Capital T, simple B, or capital T, simple B, simple B. This is the aspect of the parent. So then, this guy can't get capital 2 Bs because one parent only have simple B. So therefore it can be taken. So these are the two plants. So the finally we come into a conclusion. Plant 1 is the genotype is this, and for the plant 2 genotype is this. Got it? Without parents we can't argue now what is the final outcome of the plant 1 and 2, but with respect to the parents from this aspect, parent is having a simple double B, and here the parent is having both simple and simple B and simple T double. So therefore it cannot be capital doubles, right? Capital doubles, no? Right, okay. Now this, what are the gametes? Capital B, simple D, simple B and simple D. These are the gametes, right? Then capital B, capital T, capital B, simple T, then simple B, capital T, and simple B, simple T. These are the gametes, right? Then what are the outcomes? What are the outcomes? What is asking? The number of different genotypes, no? Right. Genotypes it is asking. Now we'll go through the one by one. Now capital B, capital B, capital T, simple T is a 1. Then capital B, simple T is a 1. Then capital B, simple B, capital T, simple T is a 1. Then capital B, simple T, simple T is a 1. Then once again, capital B, simple B, capital T, simple T is a 1. Capital B, simple T, simple T is a 1. Then simple B, capital T, simple T is a 1. Simple B, simple T, simple T is a 1. Now that is asking different types of genotypes. Now, are there any same ones? Capital B, simple B, yeah, there's a same one. This one and this one are same. Then capital B, simple B, capital T, simple T. No, these one are same, right? Okay. Are there anyone same? Are there any similar things? I don't think so. Okay, so therefore, these two is a 1. Now it is 1. Now these two is another one, different genotype. Now this is a different genotype 3. Now this is a different genotype, no? This is 3. So this is 4. This is 5 and this is 6. There are 6 different genotypes. So the answer for the 31 is 3. Tremendous question, right? It's an excellent question, right? Actually, that is very, very good question, right? You have to go through the parents to check whether what is the type of the plant, right? Without going through that, you can't say anything, right? Okay. Then we'll go to the 32nd. If right, okay, just note down this one. Take a screenshot. I think it's better. Those who have correctly take the answer as the third one, just let it in the chat. Let it in the live chat. Those who have correctly taken 3 as the answer, then you will be genius, right? If you have taken the answer for this one, then you are great, right? Because you have all the knowledge in the genetics and the process. Let it in the chat. Let it in the live chat. Very good. Great. 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 Right. Okay, then 32nd. Very good. Right. You can take a screenshot, right? Those who have incorrectly done. Actually, the theory part, the critical part is the theory, right? Okay. Not a genius. Don't bother, right? Okay. Non-genius, right? 32. If a child has type O blood and the mother is type A, then which of the following could be the blood type of child father? Child is simple lie, no. Child O means simple lie, right? Then mother is A. Mother can definitely be IA simple I, no. So A blood group, there can be capital I, capital A, capital I, capital A, O, capital I, capital A, simple I. Got it? But the child need to get both simple I's, no? In that case, mother cannot be this one, right? This genotype cannot be taken for the mother. This very simple thing. Very simple thing, right? The genetic basics, right? So therefore, then father need to be also carrying simple I. So then father can be a simple double I. It means father can be a O group, right? He can be a O. He can be a A even, right? Capital I, a simple I. He can be a A. Then he can even be a B, right? He can't be AB, right? So this is then these are the genotypes relevant to A and B because father definitely need to have simple I in go with mother's simple I to form a baby with simple double I. So the answer is A, B, O only. 32nd one. Answer is 5. 33rd. The sequence of bases on a messenger RNA. It is in the mRNA. So the sequence now just note down this. Right? Okay. 33rd. Now the mRNA is given. We are having the template strand of the DNA is asking. No? Now mRNA is here. Now this is the mRNA. No? So then the DNA which used to synthesis the mRNA need to be this, no? Thymine, then it needs to be adenine, guanine, cytosine, thymine, cytosine, adenine, then guanine, cytosine, and adenine. Now this is the DNA, no? Now template strand is this. Now we have to take the DNA of the coding strand. The coding strand is now this is the template strand, no? This is what is taken from. So the coding strand have to be adenine, then thymine, cytosine, guanine, adenine, guanine, thymine, cytosine, guanine, and thymine. So is there any answer? ATC? Yeah, ATC, GAA, GTT, CGT. Now this is very important, right? Now this is the coding strand. First one is the coding one, right? This is the coding strand, right? Of the DNA. The DNA has two strands, no? Now this is the template strand. This is the template. It's a template. Now this is the mRNA. Black one is the mRNA. Right? This is the one. So the answer for the 33rd is, answer is second one. Right? This is also a very good question. It is if someone actually unluckily in the third question it needs to be G, but mistakenly in the type setting it goes as F. If that two will be there in the answer, then you will definitely choose it as the answer, right? Okay. But it is asking about the coding strand, right? Based on the coding. This is a coding strand and this is the template strand. Template strand is used to take the mRNA. Then it is asking about the coding strand of the DNA, right? Coding means the other strand. There are two strands of a DNA, no? Coding and template. This is the mRNA synthesis from the template one, right? Okay. So it is asking of the coding. Then 34, which of the following enzyme has exonuclease activity? Exonuclease activity is found in DNA polymerase, right? For the 34th, answer is Fourth one, DNA polymerase has the exonuclease activity, right? Okay. 35th, which of the following genetic disorder caused by monosomy? Monosomy is what? Turner, no? Turner is monosomy, right? Monosomy is the Turner. In the Turner, that is monosomy. There's a decrease in one, right? Monosomy. Other, then Klinefelter down, they are trisomy, right? Sickle cell and color blindness, they are sexually transmitted diseases, right? Okay, they are not the diseases caused by the genetic mutation. Therefore, Turner is monosomy. Klinefelter and Down are trisomy. Therefore, the answer for the 35th one is 3. 36. Which one of the following IUCN categories in Sri Lanka has the highest risk of becoming extinct in the near future? That is the marble rock frog. Therefore, answer for the 36. These are raw theory knowledge, right? Not critical. Answer is 2. Right? Okay. Sri Lankan legume, then Trudia salonica, and the dusky strip jungle squirrel already they are extinct. So the marble rock frog has the highest risk of becoming extinct. So the answer for the 36 is second. These are very easy. Then 37. Now there's a matching. There's a matching. Column 1 and column 2. Biodiversity convention tells, right, that over-exploitation of bioresources. Yeah, the biodiversity convention is there for the rescue of over-exploitation of bioresources. Then for the A, we'll match that, right? We'll match. For the A, it is Q. For the A, it is Q. Then Cartagena Convention tell that conservation of biodiversity from the risk of biotechnology. That is the Cartagena. Cartagena means there's technology. Cartagena, biodiversity along with the technology, right? So therefore, for the B, it is T. For the B, it is T, right? Then, see, Kyoto Protocol stands for the global climatic changes. That is very easy, no? These are raw theory facts, right? C for S. Then D, CITES. CITE is there for the conservation of endangered species, right? For the D, that is P. For the D, that is P. Then finally, MARPOL convention is there in for the prevention of pollution by ships. That is correct. Then E, for the E, that is by ships means R. Is there any answer like that? Q, T, S, P, R. What is the answer? Q, T, R. That is the fifth one. For the 37th, answer is fifth. Answer is fifth. Right. 38. Which of the following is an educational or a scientific value of biodiversity? Regulating climate is an environmental importance. Recharging groundwater is an environmental value. Prevention of soil erosion is an environmental value. Water purification is also an environmental value. Help in disaster management is an educational value, because disasters take place, but with the knowledge you are having, with the knowledge you are taken from the education of referring to the disasters, then you can manage it, because help in disaster management comes under education, right? Answer for the 38 is fifth. 39, mycoplasma. Mycoplasma comes under the domain bacteria, right? So prokaryotes but do not contain cellulose. That is correct. Both mycoplasma and phytoplasma, they don't contain cellulose, right? 39. First one is incorrect. First one is correct, right? First is correct. We'll go to second one. Cannot grow in artificial media. They can be grown, right? That is incorrect. Third one, can be seen under, no, they can be only seen under light, not light electron microscope. Almost all are parasites of humans, but not found in plants. Which molecule is found in plants? That is the phytoplasm, not the mycoplasma, right? Do not possess any metabolic. No, they contain metabolic activities because they are living. Therefore, answer for the 39th is first. 40. Right? 40. Select the correct combination regarding the use of microorganisms in the following commercial product. Aspergillus niger is what is responsible for the cellulase, not the Aspergillus oryzae. First one incorrect. Vinegar is by the Acetobacter, not by Azotobacter. It is by Acetobacter. Insulin is by Saccharomyces cerevisiae. Streptomycin is by Streptomyces griseus, not the Orophacians, right? Retin is not done under, it means in the resource book, there are not a microorganism given under the retin. So the Thiobacillus ferroxidans is responsible for the extraction of metals. So therefore, the answer for the 40th is 3. Answer for the 40th is 3. Answer for the 40th is 3. Right. Now, from the 31 to 40, you can tell how much you have obtained. Out of 10, how much you have obtained. Right. Then tell me how much you have obtained for the 50 allocated. It's a tough, not a tough, but it's a just means somewhat not tough as well and not easy as well. It's kind of average paper, I think. Right. Okay. Now this is the first separate English medium discussion I have done in the YouTube platform, right? Now you have something to share with me. You have something you can help me, right? So after this live is ended, then the video will go under the video category. It means it's under stream category, no? So the video will be there remain in the YouTube channel, right? So you can just go there and express your idea or thought regarding the discussion. So it is not, it won't be helpful for me actually, because already I have the certain enough students for the teaching process, and I have enough students, and actually the students are those who are get benefited from this kind of stuff, because English medium rather than the Sinhala medium, English medium students are facing so much terribles, right? So much troubles, right? So because they are having, if we go to a class, they are, it means that the class fees so much higher, far more, 2 times more than in a Sinhala medium one, and all those classes are might be individual or group, then the cost is far more higher, right? Okay. So in that aspect, there are so many English medium students who are suffering from that economical crisis, as well as those guys, though they take so much of money, they don't offer such kind of a knowledge or value or those effort for the learning process, I think for the teaching process, right? So in that case, I think these kind of stuff and the way we are doing along with the Sinhala medium students, the support we are giving, I think might be a very important thing for you, because in our class, more than the Sinhala medium students, English medium students are working harder, and they are increasing their marks day by day. And those are, it means there are some students who are more doing works than the Sinhala mediums even, right? Doing the works more than the Sinhala medium. There are some English medium students, right? So in that case, I think this will be important for those students who are in the 2024, 2025, and those coming years also, to have an idea from this discussion, because prior to watching a video, we normally go through the comment section. So therefore, you can have put a comment in the comment section regarding the discussion, your expression, right? If it is bad, just comment, this is bad, this is not good, and this is rather ridiculous. Likewise, if it is good, you can share your expression or your idea regarding that. So one who is watching this video after the live streaming, then someone who has watched completely, then that student also recommend something regarding the video in the comment section. That is kind of a help I'm asking from you, right? Okay, so the discussion is right, and will end from now onwards. This is what I have to share with you. This is a paper. If someone didn't do the paper is there in the we'll put it in the comment section, the Google Drive link. You can download it and you can do the paper. Okay, so that's what I have to share with you. We'll meet you. It means we'll meet you in another discussion in near future. Definitely, we'll do some English medium only discussion prior to the A-level, right? Okay. Then till then, goodbye. Be safe. Work hard. Achieve your goals. There is no limit for good things, right? That is the simple thing I have to say, right? Okay. This is the part to the medical college. Okay. We'll wind up the session.